So I'm really happy to have with me Piotr Janas, who was born in Poland, but moved to the UK in 2014 to pursue a Bachelor in Science degree in microbiology and biochemistry at the University of Sheffield. He obtained his master's degree in infectious diseases in 2018 at the University of Edinburgh, where he is now finishing his PhD under supervision of Professor Jürgen Schwarze, working on the long-term effects of respiratory syncytial virus on lung epithelial cells. And the reason we are together is because we will be discussing his recent PLOS ONE Lab Protocol article and associated protocol in protocols.io entitled called Dispase Digestion of Murine Lungs Improves Recovery and Culture of Airway Epithelial Cells. Piotr, thank you so much for giving me the opportunity to talk about this paper.
Thank you so much for inviting me.
So to begin with, can you just briefly explain what the protocol is about, how to use it, and why it is relevant?
Yeah, absolutely. So basically, the protocol describes a novel approach for digesting murine lungs. And the focus for this digestion is to isolate as many and as viable lung epithelial cells as possible. And I routinely use this protocol to isolate lung epithelial cells for flow cytometry, for qPCR, for ex vivo culture. And I think it's relevant because as scientists keep working on the biology of the lung, it's turning out that epithelial cells are much higher importance than we initially thought. So they're not just there to carry out their respiration, but they have a lot of immune roles. They have huge, have really high importance in lung regeneration. So this is particularly evidence following the COVID-19 pandemic. And so I think there is a need for very good tools that will allow us to study these cells in detail.
So, and to help us understand the relevance of this novel protocol, can you again briefly describe what was the state of the art? So what was the way that scientists were isolating these cells and why you decided that something new was needed?
So there is a lot of different protocols out there, but what I think people mostly resort to when they want to isolate specifically lung epithelial cells in mice is they perform a dispase 2 digestion. And they usually do it between 30 minutes to 1 hour. They agitate the lung in a digestion mix and then follow it up with isolation or flow cytometry. So that's what I started with when I was starting my project. So I was digesting a murine lung for 1 hour using dispase digestion at 37 degrees.
And what is the innovation of your protocol?
So the novelty is that instead of performing the digestion for a short period of time at 37 degrees, I am digesting lungs for 20 hours, but in fridge conditions, so around 4 degrees Celsius.
So it seems obvious, but I just want to make it very clear. What in this new technique for digesting the lungs, what are the most critical steps?
So I would say that there is one one kind of major critical step, and that's actually one of the first steps. So as part of the protocol, I cannulate the trachea of mice and I inflate the lung with the digestion mix. Now, if for any reason this inflation doesn't work well, unfortunately, the digestion will not work great. And usually when I fail to inflate the lung well, I usually see that following the digestion, I'm getting similar numbers of cells as if I did the hot digestion for 1 hour.
I see. Oh, this is very interesting. I didn't know that that was like a very critical step. I would like us to talk now a little bit about how you optimize the protocol, the steps that you follow. And in particular, whether when you decided to say, okay, the hot and short is not as efficient as I want, let's try the cold and long. What other variables you try that didn't work and didn't make it to the final version of the protocol?
So I've worked on this protocol for quite a bit of time, so I've tried a lot of different things. One major thing that I tried is, and that's inspired by other protocols that I've seen out there, is very often people inflate the lung with the digestion mix and then plug it or seal it in. So I tried that, but I found that actually that was detrimental. So basically following the inflation, the lung has to be excised very quickly and put in a digestion mix. And it seems like there need to be this kind of free-flowing digestion mix between the lung and the solution. At least that's what I found out. Another thing is I try very often when performing these hot digestion at high digestions at 37 degrees, people agitate the lungs, so either shake or rotate. So I tried doing that. So I tried rotating the lung for the 20 hours at cold temperatures. And I didn't find that it worked very well. So I just resort to keeping these digestion mixes at cold temperature in the fridge, and they just stay there. And one other major thing that I tried is, as part of this protocol, I also propose, in my mind, most efficient way to isolate lung epithelial cells. And I do that by MAC sorting using magnetic beads. And this is a two-step process. And I tried several different approaches to that. The Miltenny, the brand that offers this kit, they have many different column sizes. So I tried several combinations. So I tried small columns. I tried large columns with small columns. I tried just large columns or columns that are specifically made for depletion. But in the end, I found that what I've put in the protocol, so the use of large LS column followed by smaller MS column, seemed to work best.
And I know this wasn't the objective of your protocol, or you didn't focus on this, but have you given any thought, or have you even tried to use different enzymes, different digestive enzymes? So that's one question. And the other, thinking ahead, have you also tried to transfer the protocol to a different species, and whether you think it might work or some modifications might be needed?
So the two very short answers will be no and no, unfortunately. But if I were to expand on that, firstly, I would love to try this protocol on, for example, human lung biopsy or human lung explant. So, for example, from a cancer surgery, things like that, and try to dislodge epithelial cells using this protocol. And sorry, the second question was?
It was about the enzymes. You know, you
Enzymes,
focused
yeah.
on dispase, but there are other digestive
Yeah.
enzymes. Did you try?
So unfortunately, no. This is basically, I had to pick my battles when optimizing this protocol, and I thought I'll just stick with one enzyme. But this is something definitely to improve on. Using this enzyme doesn't come without problems. So if some cell surface markers are sensitive to this enzyme, and they might get lost. So if you are interested in this specific marker that's sensitive, this might not be a protocol for you. So I would love to try other enzymes, other enzyme mixes to see if I can kind of substitute it and preserve these markers on cell surface. Yeah.
Thank you so much for the very thoughtful answer. I think is very illustrating. I would like to shift gears now, and I would like to ask you, you know, you have been working on this protocol for a very long time. You try a few things, and then you went ahead, you made it public, but then you decided to get it peer reviewed. What was the motivation to have a formal peer reviewed paper out of this protocol?
So, well, I really believe that this protocol can be helpful to many other lung epithelial researchers. And I guess putting it out there as a peer-reviewed protocol would kind of ensure that this is a high-quality work and people can trust with that this is a good approach if they want to try it.
And can you then tell us how the feedback that you received from the peer reviewers shaped the final version of your protocol?
So in our experience, the peer reviewer helped us with generally being more precise. So I guess we can thank the reviewer for making the protocol more reproducible.
And why did you decide to make those comments public, the peer review history public?
Yeah. So I'm an idealist when it comes to science, and I think we should strive for complete transparency in science. And if there were any mistakes or omissions on our parts that had to be addressed before publishing this protocol, I don't think there is anything to be ashamed of, and I didn't see any downsides.
And I know, you know, your protocol is relatively recent, so I don't know whether you have an answer for this question. But my last question to you would be, what has been the significance, or what do you think the significance will be of this peer-reviewed publication for your young career?
So this protocol already had an impact on my career because it essentially allowed me to complete my PhD. So it's the reason I started working on this is because the current protocols at the time didn't give me the tools that I needed to do my work. So I had to do this first, and it allowed me to complete my PhD on that topic. And I think it might be of significance and importance, particularly in the context of, for example, sequencing. So to date, when using other digestion methods followed by cell isolation and sequencing, we might be missing quite a lot of populations within the epithelial population. So I think this will help with enriching these rare epithelial subpopulations for these kind of techniques.
Piotr, thank you so much for this conversation. I think it's been really pleasant. Before we finalize, I just want to ask you if there's anything else that you would like to share that we haven't touched so far.
I think we've touched on everything, and I just really hope this protocol will come and will be useful to someone, because I really believe that it's really good.
Well, thank you so much.
Thank you as well.